Dr. Lanfen Chen to give. Okay, so firstly, I will give a brief introduction for the Professor Lanfen Chen. Dr. Lanfen Chen is currently a professor at the School of Life Science, Xiamen University. She obtained her bachelor degree and a master degree at the Xiamen University in 1997 and 2000 respectively, and her PhD at the Albert Einstein College of Medicine, New York in 2006. She was a research fellow at the Brigham and Women's Hospital, Harvard Medical School in 2007 to 2012. Then she became a principal investigator at the School of Life Science, Xiamen University since July 2012. The Hippo pathways has emerged as a critical developmental pathway, contributing to process the regulating tissue growth and organ size. And MST1 is the mammalian ortholog of the Hippo kinases. So Lanfen's lab aims to characterize the regulation of physiological functions of the MST1 and MST2 protein kinases in the immune system, especially in the innate immunity. So let's welcome Lanfen. Okay, Lanfen, please.
Okay, thank you, Jim. So let me stop my video. Okay, good morning, everyone. So first, thanks, Jim, the nice introduction. And also, I would like to thank Xiaolei and Chuang Wu for organizing this nice weekly immunology online seminar. So today, it's my pleasure to present our recent work here, function of hyposignaling in macrophages and host antimicrobial defense. Let me briefly introduce the hyposignaling pathway. And this pathway was first identified in Drosophila to control organ size. It's an evolutionary conserved pathway. Every component of this pathway has been identified in mammals. So generally speaking, MST1 kinase are the orthologs of Drosophila hippokinase are core kinase in this pathway. And MST1 and 2 kinase first phosphorylate the downstream substrate and eventually inhibit the transcriptional core activator YAP and TAZ's function to control cell growth and death. And several studies have shown that loss of hyposignaling pathway will lead to the organ size enlargement and eventually tumor development. So the hyposignaling pathway was first considered as a tumor suppressor pathway. And collaborating with Dr. Dawan Zhou, we found that actually knocking out of MST1 kinase or overexpression of YAP will lead to enlarged liver and eventually liver cancer. In contrast, knocking out of YAP will result in liver cell death or liver failure. And this result demonstrates that hyposignaling plays a very important role to maintain the tissue homeostasis. And my lab is interested in the function of hyposignaling pathway. Previously, we reported that MST1 and 2 kinase are highly expressed in lymphoid tissues, such as thymus, spleen, and lymph node. And knocking out of MST1 kinase in hepatopoietic lineage, the mice exhibit T and B cell lymphopenia and recurrent bacterial and virus infection, as well as the spontaneous autoimmune disease. And in 2012, 2 groups reported the loss of function MST1 mutation in human patients. Those patients have a primary immunodeficiency syndrome, and the clinical symptoms include the clinical symptoms including T and B cell lymphopenia, neutropenia, and also the patient are easy to get the bacteria or virus infection, and also some autoimmune disease. All these mice and humans evidence suggested that MST1 and MST2 kinase have important function in regulating both innate and adaptive immune response. So when I back to China to set up my own lab. So regarding the T cell immunity, we found that actually MST1-2 kinase is critical for maintaining the naive T cell is critical for maintaining naive T cell stature and also can inhibit the T cell activation. And we also found that MST1-2 kinase and its downstream transcription co-activated test can regulate the differentiation of T helper 17 and T regulatory cells. So hyposignaling it's important for balancing the inflammation and immune tolerance. And regarding the innate immune cells, we reported that MST1-2 kinase can regulate the phagosomal and mitochondrial ROS production for killing the bacteria during the infection. We also find that MST1-2 kinase can regulate antioxidant defense. And so this pathway is important for the redox homeostasis in the innate immune cells. And today I'm going to talk about the function of hyposignaling in innate immunity regarding the infection, host defense, and redox homeostasis. We reported earlier that loss of MST1-2 kinase in hematopoietic cell lineage, the mice easily cause spontaneous infections such as pneumonia. And then we found that when in the the MST1-2 kinase innate cell conditioning knockout mice are more susceptible to the cervical ligation punch-induced bacterial sepsis. And further study suggests that MST1-2 kinase deficient bone marrow-derived macrophage cell have defect in phagocytosis and bacteria killing activity. So this results suggest that MST1-2 kinase are important for control infection in innate cells. And previous studies have shown that the ROS induction, including the mitochondrial ROS and the phagosomal NADPH oxidase complex producing ROS are critical for killing the engraft bacteria during the infection in these macrophages. And then we found that actually upon the bacterial infections such as E. coli or Listeria, the mitochondrial ROS or total cellular ROS can be induced in the wild type bone marrow derived macrophage or neutrophil. You can see with the ROS diet shift, but we cannot see this phenomenon in the MST1-2 knockout deficient phagocytes. And we also find that MST1-2 kinase are required for the cytoskeleton remodeling in the macrophages, and so it's critical for the mitochondrial recruitment to the phagosome. So from this figure, you can see in wild type cells, when the cell will incubate with LPS-coded bits, you can clearly see the mitochondria is recruited and wrapped on those bits. But in the MST1-2 knockout cells, we could not see this recruitment. And to make the long story short, by using the biochemistry and genetics procedure, we found that actually during the infection, MST1-2 kinase directly phosphorate the PKC alpha, and then activate the small GTPase REC1, which promote the assembling of ECIT and chapsticks to recruit the mitochondria to the phagosome and produce ROS. And on the other hand, the small GTPase REC1, also an important component of NADPH oxidase complex, is required for the phagosomal ROS production. So both these will increase the ROS level and then kill the engraft bacteria. And we also found that with the toll-like receptor ligands treatment, especially the toll-like receptor 1, 2, and 4, and this signaling can activate MST1-2 kinase, as shown by the increased phosphorylation level of MOB1, which is physiological substrate of the MST1/2 kinase. So all the later work, we were using the phosphorylation level of MOB to indicate the activation of MST1/2 kinase. And this process actually is MyD88 dependent. But actually, we know that the toll-like receptor can activate the MST1/2 kinase. But how it can be activated, it can be activated, it's still not very clear. And recently, we found that actually extracellular mechanical force might have a role in the toll-like receptor mediated MST1/2 kinase activation. Okay, and many studies have shown that mechanical force can regulate hyposignalings. And for the epithelial cells, actually, one cell will cultured in the ragged parus on the stiff matches, all the cell were stretched, will regulate the hyposignaling to promote the YAP and TAZ, the nuclear translocation you can see here, and then activate YAP and TAZ and promote cell proliferation. So this suggests that mechanical force can regulate the hyposignaling pathway. And for macrophages, actually, mechanical force can also regulate macrophage's function. As we know that the mechanical environment of macrophages can vary from the soft to stiff in the environment, especially during the inflammation. And interestingly, we found actually this extracellular matrix stiffness can modulate the macrophages phagocytosis and bactericidal activity during the infection. So we did bacteria killing assays by conditioning those macrophages on different softness and different softness of the matches, such as the 5 kPa, 35 kPa or glass. So at the beginning, this intracellular bacterial membrane indicate like how many bacteria will engraft. So we can see actually in the softer matches, such as 5 kPa, the phagocytosis activity is very low. So you can see fewer bacteria will engraft in this macrophage cultured on the softer matches than that of the stiffer matches. And one time last, actually we can see, interestingly, actually the bacterial killing activity is lower, and also all the intracellular bacteria number is higher in these softer matches, the culture, the macrophage, compared to the those on the glass matches. So this indicated that extracellular matrix stiffness is essential for macrophage phagocytosis and bactericidal activity. So we asked what receptor on macrophage can sense the mechanical force and regulates its function. And there are several mechanical sensitive channels were discovered in the eukaryotes, including the sodium channel, DEG subfamily, or some TRP receptors, or the potassium channel like TREK1. And also recently found that the piezo family, including the piezo 1 and piezo 2, which are calcium channel. So to study the function of the mechanical sensitive ion channel in the macrophage mediated innate immunity. So first we determine the expression level of mechanical sensitive ion channel on the macrophage, and found that only piezo 1, but not piezo 2 or other reported mechanical sensitive ion channels were highly expressed in the macrophages. And then we confirmed that it's piezo 1, but not piezo 2 are expressed on the myeloid immune cells, as by using the TD tomato red tagged piezo 1 protein, or the GFP tagged piezo 2 protein reporting mice. So we can see here that myeloid progenitor isolated from the bone marrow, all the DC cells, macrophages, and granulocytes are all highly expressed the PISO1, but not the PISO2. So green line is PISO2. And then and furthermore, we found that actually lack of PISO1 abolished the enhanced phagocytosis or bacterial killing on the stiffer culture matches. So suggesting that PISO1 receptor is involved in mechanical force mediated bacteria engulfment and killing. And then we check the in vivo bacterial killing activity. And as we expected that the cyclical ligation and punch induced bacterial sepsis killed more PISO1 conditioning, innate cell conditioning knockout mice. And we also can detect higher bacterial load in multiple organs such as spleen, lung, liver, and kidney of the PISO1 deficient mice upon CLP induction. Similarly, we found that PISO1 deficient macrophage also have defect of bacterial killing and phagocytosis, as shown by this shift of the engraft of the Fiji labeled E. coli, or the defect on the intracellular bacterial killing. So this result indicated that both the proper extracellular matrix stiffness and PISO receptor are important for macrophage phagocytosis and bactericidal activity. And then to characterize the law of PISO1 in innate immune cells upon infection. So we prepare the bone marrow derived macrophage from the TD tomato tagged PISO1 transgenic mice, and then incubate them with the LPS coated beads, or infect with the BFP labeled E. coli. And clearly we can see that actually subcellular PISO1 condensed and wrapped on these LPS coated beads. All the condensed and colocalized with the bacteria. And these phenomena cannot be found in the toll-like receptor knockout cells. So this suggests that with the infection, we can say that the bacteria or beads, the ligation, we can the toll-like receptor can recruit the PISO1 associated with PISO1 and might trigger the downstream signaling. And interestingly, when we treated the cell with the LPS, so it's a soluble toll-like receptor ligands, we also can see the association of PISO1 and toll-like receptor 4. So as shown by this structure illumination microscopy, we can see that actually here without the in control cells, there's red and green dots is completely separate. But upon the LPS treatment, you can see the yellow dots and some of the association particle are actually enlarged. And this association was confirmed by the immunoprecipitation, as you can see that with LPS treatment will promote the toll-like receptor binding to the PISO1. So this suggests that not just because bacteria you can see is a particle, not just upon the particle touch to the cell, the soluble ligands trigger the toll-like receptor signaling can also trigger the can also associate recruit the PISO1 and associate with toll-like receptor. And then our further study show that actually PISO1 is it can also regulate cytoskeleton reorganization. Upon YODA1 treatment, which is a PISO1 agonist, or treated with LPS or infected with the E. coli, we can clearly see the philopodia formation on those cells. And also upon those treatment, we can see the cellular stiffness increased in those treated cells. And PISO1 receptor is required for this phenomenon, as we could not see the effects on the PISO1 knockout macrophages. And we have previously have shown that cytoskeleton remodeling is also important for infection-induced ROS production and bacterial killing. So indeed, we found that upon YODA1 treatment, so the mitochondrial ROS level increased. And we already showed that with LPS or E. coli, the ROS level also increased in these macrophages. But when we knock out of PISO1, so these increasement are all, so the ROS are all diminished on those knockout cells. And similarly, the YODA1 can enhance the bacteria killing activity of macrophages. And this effect also diminished in the PISO1 knockout cells. And also, many studies have shown that cytoskeleton remodeling is important for, okay. Sorry, okay. And then we treated the cells with the cytoclastin D. So it's a cell permeable and potent inhibitor of actin polymerization. So actually with the CYTD treatment, we can see that the philopodia formation induced by the YODA1 disappeared. And also the YODA1 induced the cell stiffness the cell stiffness were also diminished with the CYTD treatment. And similarly, it can also block the YODA1 induced ROS production or the bactericidal activity. So this result suggests that cytoskeleton reorganization is required for PISO1 receptor-mediated anti-infection activity in the macrophages. And many studies have shown that small GTPase activation involved in the cytoskeleton reorganization and philopodia formation. So we determined the activation status of the representative molecules of the small GTPase families, such as the low RAC and CDC42 family, also the RAS family, including the RAS and RAP2, and ARF family, including the ARF6 in macrophages. And we found that actually upon the YODA1 stimulation, only RAC1 can be activated in the YODA1 treated macrophages. And then we confirm that YODA1 treatment can increase the RAC1 activity in the wild type macrophages. But this phenomenon could not be seen in the PISO1 knockout cells. And similarly, we found that actually YODA1 induced mitochondrial or cellular ROS increasement can only be seen in the wild type cell, but not in the RAC1 knockout cells. And also YODA1 induced bacterial acid activity, enhancement of the bactericidal activity could not be seen in the RAC1 knockout macrophages. So combined our previous study this data suggests that REC1 plays as a downstream effector of piezo-1 and toll-like receptor signaling to regulate RS induction and bactericidal activity in macrophages. As we just mentioned, that toll-like receptor signaling activate hyposignaling, as shown by the activation of MST1/2 kinase and regulate the REC1 activity for downstream bactericidal activity. So we asked, we were wondering whether piezo-1 mediated macrophage bactericidal activity through the MST1/2 signaling. And indeed, we observed UDA1 treatment can increase the phosphorylation levels of MOB, but not in the piezo-1 knockout cells. And piezo-1 knockout cells and knocking out of piezo-1 in the macrophages can abolish the LPS-induced phosphorylation of MOB. And we also confirmed that UDA1 induced activation of REC can only be seen in the wild type cells, but not in the MST1/2 knockout cells. And also, we know that UDA1 treated can induce the phylopodia formation in the wild type macrophages, but we could not see that in cells treated with the MST1/2 kinase inhibitor XMUMP1 compounds. Also cannot be seen in the knockout MST1/2 knockout macrophages. And UDA1 induced cell stiffness cannot be seen in the MST1/2 knockout macrophages. And also we found that MST1/2 kinase are required for the piezo-1 mediated RS production, as shown here, like UDA1 induced RS production could not be seen in the MST1/2 knockout cells. And also for the bactericidal activity, it also blocked in the MST1/2 knockout cells. And then we asked how actually the piezo-1 activate MST1/2 kinase. So as you already seen here, that as the ion channel, piezo-1 facilitated calcium influx into the cytoplasma. So with UDA1 treatment, we can see the rapid calcium influx in the wild-type macrophage, but not in the piezo-1 knockout cells. And interestingly, LPS-induced calcium influx was also dramatically suppressed in the piezo-1 deficient macrophages, indicating that ion channel piezo-1 is important. The calcium ion channel piezo-1 is important for induction of this calcium influx by toll-like receptor signaling. We then found that actually without the extracellular calcium, UDA1 could not induce the MST1/2 activation, as shown by there's no increase of phosphorylation level of MOB1. And when we treated the cell with the calcium cluster BAPTA, we also find that UDA1 induced the increasement of the phosphorylation level of MOB1 were diminished. Okay. So this result suggests that toll-like receptor triggered piezo-1 activation can increase the calcium influx for activation of MST1/2 kinase. And then we thought to determine how calcium mediated MST1/2 kinase activation. So we treated the cell with inhibitors of several calcium-dependent kinases, such as PKC, FAK, and comodulin kinase. We found that actually only comodulin kinase inhibitor can block the UDA1 induced phosphorylation of MOB1. And our further studies show that Commodulin kinase 2 can directly bind to MST1 and also can directly phosphorate MST1. And when we treated the cell with the Commodulin kinase 2 specific inhibitor TFA, it can also block the YODA1 induced RS production and also block the YODA1 induced enhancement of bactericidal activity. So this result showed that upon infection, toll-like receptor and associated with PSO1 and increase the calcium influx to activate Commodulin kinase 2 and then directly phosphorate MST1-2 kinase to activate MST1-2 kinase for following activity. And so taken together, we found that actually upon infection, so with the toll-like receptor signaling, it can associate with the PSO1 and activate PSO1 to increase the calcium influx and activate Commodulin kinase 2, and then enhance the MST1-2 activity, and eventually activate a small GTPase RAC1 for cytoskeleton remodeling for phagocytosis and bacterial clearance. And my last part of the talk will be how MST1-2 maintain the redox homeostasis in phagocytosis during the infection. As everyone knows, that actually RS actually everyone knows RS have a double-edged sword effects. Upon infection, increase the RS is critical for destroying the passenger inside the cells. But increase the RS can also cause the oxidative damage and lead to the cell oxidative stress and eventually cell senescence or age-related disease. And when we analyze the RS level in the macrophages, we found that it's an interesting phenomenon. The basal level of RS level actually the basal levels of RS actually were increased in the MST1-2 knockout cells. So we wondering whether MST1-2 kinase play a role in regulating RS level in macrophages. And when we analyze those cells, and we found that actually in the MST1-2 knockout macrophages, the protein carboxylation level levels are increased, and also the apoptotic events are also increased in those MST1-2 kinase deficient macrophages. And with NAC treatment, the protein carboxylation level or the apoptotic events will be decreased. So it means that these MST1-2 deficient macrophage undergoing the oxidative stress. And then we also observed that more granules of lipofuscin, a cell aging marker, lipofuscin, actually, like you can see these black dots under the electronic microscopy. So there are more granules of lipofuscin in the younger MST1-2 deficient macrophage when compared to the similar age of wild type cells. And also we found that the antigen presenting activity of macrophage was also decreased in earlier age of MST1-2 deficient macrophage when compared to the similar age of wild type cells. And then our further, so it means that actually loss of MST1-2 kinase will lead to cell senescence and premature aging. And then we found that actually upon the infection, when we infect the cell with the bacteria E. coli, the GFP labeled E. coli, we can clearly see that MST1-2 kinase can be recruited and associated with this engrafted E. coli. So we consider as a phagosome. And with NAC treatment, and this we could not see this improvement. So it means it depends on RS production. And also E. coli induced MST1/2 kinase activation shown by the increased phosphorylation level of MOB can also will also diminish in cells treated with NAC. So these two results suggest that MST1/2 kinase might sense the phagosomal and mitochondrial ROS, and also release the ROS can activate the MST1/2 kinase in macrophages. So actually, for regulating the ROS level in cells, the induction of antioxidant enzyme is important for protecting cell against the oxidative stress. So we measure the mRNA. Level of several antioxidant genes as shown here. Then we found that actually the expression level of those antioxidant genes were significantly decreased in the MST1/2 deficient macrophages. And it has been reported that NRF2 protein is a major transcription factor for induction of antioxidative enzyme expression in response to the oxidative stress. And NRF2 protein level is regulated by the Hippo-1 NRF2 regulation system. As shown in this cartoon, actually when cell is in unstressed condition, the Hippo-1 actually can form the dimer or polymers and bind to the NRF2 and E3 ligase. So promote the NRF2 ubiquitination and degradation. So the basal level of NRF2 are very low in the unstressed condition, unstressed cells. But when the cell is under the oxidative or the electrophilic stress, the Hippo-1 dimer or polymers are will be dissociated, and then the NRF2, the ubiquitination level of NRF2 decreased, and then there's more NRF2 protein translocated to the nucleus and trigger the downstream antioxidant enzyme genes expression. And we define that actually the higher ubiquitination level of NRF2 and lower the total NRF2 protein level in this MST1/2 knockout suggests that NRF2 might involved in this antioxidant process in the MST1/2 knockout cells. And our further study found that actually, interestingly, we use MST2 to show the result. So actually, MST2 can directly bind to the Hippo-1 and also can phosphorate Hippo-1, directly phosphorate Hippo-1. And fortunately, we identified that 4 phosphorylation sites on the Hippo-1 mediated by the MST1/2 kinase. So these 4 phosphorylation sites are very close to the BTB domain of the Hippo-1, which is important for Hippo-1 homodimerization and or association with the E3 ligase CUL3. So to study the MST1/2 mediated phosphorylation of Hippo-1, what the function of this MST1/2 mediated phosphorylation of Hippo-1. So we generate 2 mutant, Hippo-1/4A and Hippo-1/4D. 4A is actually a non-phosphorylatable mutant of Hippo-1, and 4D actually is constitutive phosphomimic mutant, indicate that the MST1/2 kinase activity is maximum. And then we transfer, then we transfect the cells with different labeled and different mutant formed of Hippo-1. And then it's clearly our results show that Hippo-1/4A, so it means there's no MST1/2 kinase activity. We can see the Hippo-1 actually 4A formed large and completely overlaid big four size, it means that it formed the polymers or dimers. And 4A can also, itself can also form the large stop. And then if it's co-transfer with the wild type, as we know that wild type, it could be phosphorate or could be no phosphorate. So you can see there's some are large dots and some are small dots. So, and then some are colocalized with this 4A form. But for the KEAP1 4D, we see it's much more diffused the dots on these cells. And it's completely excluded from the 4A dots. So it means that constitutive phosphoraminic mutant could not form the polymer. So it suggests that MST1 kinase can phosphorate KEAP1 and prevent its polymerization. And then we study the KEAP1 mediate the NRF2 protein stabilization. So when the cell was treated with the CHX, so we can see wild type of KEAP1 4A overexpressing cells, we can see that actually NRF2, the protein level of NRF2 dropped very rapidly dropped in like 3 and 6 hours. But when the cell were overexpressed with the KEAP1 4D, we can see actually the NRF2 protein level lasts a very long time. So it means that, so from this cartoon, we can say that MST1 kinase can phosphorate KEAP1 and dissociate the KEAP1, prevent its prevent the polymerization, prevent its polymerization. So will decrease the NRF2 ubiquitination and increase the NRF2 protein level in the cells and trigger the downstream antioxidant gene expression. So, I will summarize this part of work here. So we think upon the infection or during the regular respiratory activity in the mitochondria, the RS, increase the RS will directly activate MST1 kinase and then phosphorate downstream KEAP1 and prevent its polymerization, and then increase the NRF2 levels, NRF2 level, and promote the downstream antioxidant genes. And here I'm going to summarize today's talk. So actually, our studies show that MST1 kinase are critical for maintaining the redox homeostasis in macrophage during the infection. So I will go through these two processes again here. So upon the infection, toll-like receptor associated with the mechanical force receptor piezo1 and trigger the downstream MST1 kinase and RAC1 activation and RAC1 signaling to regulate the coordination of mitochondria and flexon to produce a lot of RS for pathogen killing. And at the same time, the increased RS will further activate MST1 kinase and trigger the NRF2 mediated antioxidant activity to prevent macrophages from oxidative damage and aging. So I will stop here. And last, I'm going to, to thank the lab members in our lab. It's especially Genjing and Yilang and Zhao Hao and Junhong Jingjia, and also my colleagues in the Xiamen University. And I also would like to thank the Dr. Chung Wu and Dr. Yu Jiesun from Peking University for helping us using the atomic power microscope to measure the cell stiffness and also measure the matches, the different softness of matches. And also I would like to thank Dr. Zhao Songyu and Dr. Kun Yijian from Chang'an University for helping us set up the mass spec activity assays. And some mice will come from the Dr. Randy Jensen from MD Anderson Cancer Center, and also some from the Dr. Avaruk of the Harvard Medical Schools. And I also would like to thank the funding support from the National Key R&D Program of China and the National Natural Science Foundation of China, and also some fund from the State Key Laboratory of Cell Bio of the stress cell biology, and from the university. And then thank you, everyone. I would like to take some questions.
Right. Great talk. Thanks for the interesting and also very, very comprehensive stories presented by Lanfen. So Rongze Oliver Lu has a question. So great talk. The first one is the interaction between piezo-1 and toll-like receptor depends on the mechanical cues. The second one is, does the effect of piezo-1 and MST1 and 2 on bacterial clearance depends on YAP?
Okay, so thank you for the question. So first, actually, I specifically mentioned the LPS treatment. So I think that means that not just if you're thinking about beads and bacteria, it's solid, so it press the cells will trigger the piezo-1 activation. But when we use soluble ligands, so like LPS, and we also use like the LTA for TLR2, they also can trigger the toll-like receptor association with piezo-1. So I mean, and that means that not just that mechanical cue, but also like signaling, like directly like a toll-like receptor, the signaling. I don't know how. It probably the receptor change something, the conformation. It will recruit the piezo-1 and then trigger its activation. So it's very interesting. So we actually try to figure out how the actual piezo-1. It's not just based on the matrix pressure. And the second question, I think, for YAP, I don't really think it's depending on the YAP, because the YAP expression level is very low in the macrophages. And then I think the Hongyan one from the Shanghai, the Senhua Shuo, Hongyan actually reported that upon some it's really induced a very late point, not really at the beginning. It probably in response to some inflammatory cytokine stimulation, the YAP can be increased, and then it can regulate that. I think it's M1, M2 differentiation. But for this study, the process, I don't think the YAP is involved. Yeah, thank you.
Okay, so is Paul Huang, you have a question, right? So can you unmute yourself?
Hello.
Hi.
Hello. Thank you so much. It's a very nice talk. It's just very exciting finding. I have two questions. The first question is that as I recall, recently people also showed that the macrophage can be also invaded by the sera, for example, through toll-like receptor 3 pathway. But at the same time, macrophage can do phagocytosis to clear the infection bacteria. So I'm just trying to figure out, for example, toll-like receptor 3 pathway versus your toll-like receptor 4 pathway through this signaling. Can you comment about it, the specificity, and then in terms of the function of different tolls, including the phagocytosis and infection?
Okay. So sorry, I just wondering, because cannot remember toll-like receptor 3. It's surface or like endosurface, it's located in intracellular. I cannot remember toll-like receptor 3, but okay. But actually, our study, I'm not talking about the toll-like receptor 3. Actually, our study, including our previous the RS induction study, MCO mediated interaction. Actually, we found that actually these effects only for the surface toll-like receptor. As I mentioned, the toll-like receptor as I mentioned, the toll-like receptor 1, 2, and 4, but I cannot remember toll-like receptor 3. But for the toll-like receptor 7, 8, and 9, actually it's more like an intracellular, located in the intracellular membrane. So actually it does not have this effect. And we also check whether the piezo-1 can co-localize or associate with a different toll-like receptor. We found actually it can co-localize with the toll-like receptor 2 and 4, but not with the toll-like receptor 9. I don't know about toll-like receptor 3, but I will find out what actually the ligand and the location of the toll-like receptor 3. I'm sorry, I cannot give more comments for the toll-like receptor 3. But yeah.
Okay. Yeah, thank you. I'm just thinking that, in vivo condition, the macrophage can be infected, right? But at the same time, it can sense the LPS. That's just make me feel like, what's about the specificity. And then the second question is that, macrophage exists in different part of the body, including the liver, peritoneum, right? And then they can be all regulated, depends on different transcription factors, right?
Yeah.
What do you think about this signaling pathway during the infection in different part of the body? And the reason why I'm asking is that if there's a specificity, or maybe there's no specificity, and then based on your data, how about, for example, neutrophil function, the signaling pathway in the neutrophil, and also in dendritic cell, because you show that there's a defective antigen presentation in macrophage, but the dendritic cell should be the most professional cells to present antigen, right?
Yeah, actually, I don't know whether I can answer your question all of them, but actually, let me just do what. Actually, we didn't check the dendritic cells, but I think maybe okay. And then for neutrophil, I think some function probably will be similar, because we found that when MST1 mediated RS production, it's quite similar. From our work showing that it's very similar in the macrophage and neutrophils. And then for the different those macrophage in different tissues, actually I do think it probably have different function, but basically piezo-1 is very important for those cells in response to infections. Actually, last year the Richard Farrell group published also a work like I think he also mentioned about macrophage and neutrophil. It's in the lung during the infection, so it's more like because lung it's like the pressure. It's called the psychological force. I don't know whether my pronunciation. The psychological like hydrostatic pressure. So it's kind of like more this kind of pressure can activate the piezo-1. And their work show that it's through the transcription factor C-zone and later regulate the HIF1 alpha transcription factor, and then for producing the IL-1 beta and CXCL2. So like CXCL2, you can recruit more other like inflammatory cells. So this is like their work shows that. But I think that recently, like more and more work have been published talking about that piezo-1 is very important in the macrophage function. And actually last month, I think also in Nature Communication, there's one paper showing that piezo-1 function in the macrophage is actually, I think their work just showing that it's very important for the cytoskeleton organization for actin polymerization. And it's required for sensing the if you like now like the nanoparticles particles, okay. So like if you implant the nanoparticles to the bodies, so macrophage have to sense that to take that. So it's saying like piezo-1 is required for this process. It means that it's required for the phagocytosis. I think, and then I don't know, and then this group's work is showing that actually it depends on the kappa B activity and STAT6 activity. So I think now it's piezo-1 mediate those functions in macrophage or neutrophil. It's just at the beginning. So every group like gave it's their own explanation and their own interested pathways. And talking about the different tissues, I think that PISO1, here we just showed that actually, because when we try to submit our paper, that Dr. Rachel Ferrer's Nature paper just came out, so it totally destroyed our study. So we really modified a lot of our data. So at the beginning, we have to get rid of some overlap data and then try to make our own story like interesting. And actually, like for PISO1's function, it's a major function. It's like still is a mechanical pressure, and also the fluid share like the power like in the blood flow and also urine flow. So it's compress the PISO1. So I think in different tissue that the stiffness are different, and also the environment are different. So for macrophages, that really it's in very completely different environments. So how the PISO1 can sense this environment, and also how it responds to the infection triggered, like what we just say, the LPS triggered toll-like reset signaling. I think it really depends. It will be case by case. It's more specific. Yeah, that's my answer. I don't know whether I answer all your questions, but
yeah, I really like the idea that PISO1 and then extracellular matrix and an infection in peritoneum, for example, you know, a lot of cells, including macrophage, actually they are floating, but some of them actually stick to the peritoneum, mesothelium, for example. But so also I really like the idea that you're proposing. It's very wonderful. Thank you.
Yeah, thank you.
Okay, Xu Zhou, yeah, you could ask your question.
Oh, I think Xiaolei and Wu Chuan, their hands were up before me. I can wait for my turn.
Well, I think
okay,
you just go ahead, and then Chen can ask, and I will ask after Chen. Yeah.
Okay, sorry.
Yeah, I have I guess it's really interesting that the second part was the keep one modification part. And I remember once your gel slide shows that when you add NAC, you do see MST1 phosphorylation, but you lose the capability of I forgot the exact assays, but I remember you still see MST1 phosphorylation, and then also phosphorylation on keep one tend to activate in RF2. And do you think the function of MST1 on keep one is ROS independent or dependent?
I'm sorry, I didn't really get your question. So
right, just the activation of RF2 through MST1 phosphorylation, whether that also depends on ROS.
ROS, yeah. So yes, I think it depends on ROS, because when we treated with NAC, actually totally block all the process. But I think your question is very good, because before someone also asked whether what time and what state of the what time and what place the MST1 kinase were activated by the ROS. But I think that I was thinking about this question. It's difficult to say, but I think that it's interesting. Actually, MST1 I really feel like it's complete. It's like a very general distributed in the whole cells. So you can detect it on the membrane and cytoplasma, even in nuclear. And then when you do the staining of MST1, so you can see everywhere. But once you treated the cells with certain stimulators, including the ROS, and we can see the condensed dots. So I really feel like somehow it triggers some signaling, and then gathered that local distributed MST1, and then have the function. So that's we always see the staining pattern, like when you don't have any treatment, like you can see the totally smear distributed of MST1, and then any kind of treatment, you can see the dots, like larger dots. So I was thinking probably like ROS somehow like include the local MST1 kinase, and then have the function to trigger like that. And then whether like toll-like receptor calcium signaling directly trigger the MST1 kinase, whether the RS trigger. I think that probably is your question. I think it probably just the location depends on, and timing, because the RS, it seems that it have to be a little bit late, and then later it further activate MST1 kinase. Yeah.
You have a follow-up question after that. So it's nice you show that baseline in the MST1 double knockout macrophages, you see elevation for ROS. So that's indicating that at baseline, there's a fair level of ROS production. Right. Since the activation of RF2 depends on ROS that regulate MST1 and 2. Yeah. And do you know where do those ROS come from at baseline, and even in the resting state in macrophages?
Right, I think it's from the respiratory chain, from mitochondria, because I didn't really show all the data for this. This paper was published in 2019, so I didn't really show all the data, because actually we also show the mitochondria RS, even without the infection, if we just block the respiratory chain and to increase the RS, and we also can see the similar phenomena. So I think here I just used during the infection and what happened. But actually basal level, if the cell is doing the respiratory reaction, it can continuing generate the basal level RS, and it could be accumulated. And if this RS could not be clearance, could not be cleared immediately, it will cause some oxidative stress. So I think for our work, I think it's very important, MST1 kinase, it might have this important, to maintaining the basal level RS function. So it always watching whether the RS level is too high in this resting cells, and once it's too high, okay, it can to using this to regulate Hippo1 and NRF2 and to generate some antioxidant enzyme to clear it. And once it's RS is low, so MST1 kinase activity is blocked. So I didn't really show here, but we actually did this. We have those data to spot that. Yeah.
Yeah, that's really, really interesting. I just wonder if there's anything known about in the double knockout mice, especially in myeloid knockout mice. What's the phenotype, the known phenotypes?
What double knockout? I'm sorry, I cannot.
Sorry, MST1 and 2 double knockout myeloid knockout mice. What's the phenotype relating to infections, or even at resting state?
I think it's compared to the Valkyrie, actually the phenotype it's mild, but when the mice it did easily cause the infection. Yeah, for the myeloid condition knockout. But MST1 kinase, the general knockout is lethal. So for myeloid, yeah, it's more I think it's really easier to call infection.
Thanks.
Yeah, thank you.
Okay, next one is Xiamen. Sorry, I missed your science. Is Xiamen here?
Yes, no problem. Hey, Lanfen, this is a really beautiful talk. So I really like your work. So I think my question is also, I think, related to the one that Olivia asked at the beginning. So I think the model here is once the bacteria or the LPS bind to TLR4, it associates with piezo1, and then trigger the calcium signaling and the downstream MST1, 2. So I just wonder, where does the tissue stiffness play a role in this model? If that means the tissue stiffness, the stiff tissue can independently activate piezo1, in parallel to the TLR4 associated activation.
I think. Oh, I'm sorry. I think it's I think it's difficult to really separate this process because it definitely like for macrophage, if you want to engraft or want to respond to some stimulation such as LPS, it really like a macrophage need to grab on. I don't know whether I'm right or wrong. That's what I think. Need to grab on some place, and if it's too soft, it seems like it could not really hold the cells, and then we found that actually in our data we show that actually the cellular stiffness, it's really so the cellular stiffness is correlated to the F-actin polymerization. So the cell like it need to hold its shape or to like a stiffer, like you have to if you want to grab something, you need to be stiffer. If you're too soft, you could not really grab anything. So I think if we really could not separate the mechanical like matches force and to the signaling, I think it could be a combined effect, because I think I think Huang totally block the name. So it showed that if the cell is too soft, it also have effect to kill the tumor and also to kill the bacteria. So it really
I think that's Huang Bo.
Right. Sorry.
Yeah.
Also, Huang Bo just show that. I think it's very similar. Like you have to make it yourself like stiffer, and then this stiffer in our data we show that actually piezo 1 it's required, because if without piezo 1, that F-actin polymerization is totally disrupted. It could not really stiff. Yeah. So I think
I see.
all the
Okay.
infection is the same. Yeah. Huang Bo, right? Huang Bo 老师. Yeah. Sorry.
Yeah. So I have a technical question. So I remember in one of the slides you showed the localization of piezo-1 and the TLR4. So they seem to be like everywhere in the cytoplasm. Is that right? Or I didn't get that.
Yes, I think piezo 1 it can all be it can all be also localized in intracellular membrane compartments. So it's not just on the surface. Yeah.
I see. Okay. So that's done by like an antibody staining, right? Or it's a
No. It's you mean this figure, right?
Yeah. Oh, these are RFP. Okay.
Right. It's that so actually we use this piezo 1, the tomato tag, the transgenic mice, and so you can directly check the RFP signal. So it's very good, because piezo 1 is a huge huge protein. So it's I think it's like 900 kilodalton. So it's very difficult to to do other manipulation.
Yes. I see. That's very cool. So so the other question I have is like how does the TLR4 be turned down like after activation? Because for a typical signaling pathway, it has like the turning on phase, but also has been turning down. Is TLR4 also like endocytosed after a while?
I don't know. I didn't really look at that, but I think
I can answer
right.
this question.
Yeah,
Okay.
you're right. Yeah, it's always like that's what I always believe. Like any immune cells, once it's activated, you need to have some mechanism to turn it off. That's very important. But I don't know whether it's really internalized or it will
No,
drop.
TOR4 can be internalized.
Right. Okay. But I didn't really check for that. Yeah, in our study. Yeah.
Of course, a related interesting question might be like, where does piezo-1 go? Because it looks like the piezo-1 are colocalized in these puncta with TLR4.
Yes.
And this is like, I assume after like how long? It's after like 10 minutes or?
I think usually, I feel like when we did that, at least I don't know, I cannot remember, but usually we the time point we check will be 30 minutes, 1 hour, 3 hours, like that.
I see.
Yeah, but I don't know. But yeah, it's a very good question. But I really don't we really didn't think about how the actually pizza one where and how and where it goes. Sorry. No.
Yeah, it might be interesting, like during your time
Right.
course, like to co-stain with some endosome marker, like these early endosome EEA1 or like a late endosome marker, or even lysosome marker.
Yeah, okay. It will be just a look on these receptors where it go. Now we actually only check like the downstream signaling. Then we for the endocytosis, we only check on like where the bacteria goes. But we really didn't check whether those surface receptor when we don't know. Sorry.
Okay, that's great. Yeah, cool. Yeah, thanks so much.
Thank you.
Okay, then Chuan Wu, please.
Thanks, Lanfen, for the nice talk. I think one question I have is following Xiaolei's question. So first of all, TOFO receptor actually, as far as actually interact with the LPS, it will be internalized within 1 minute. I'm surprised you see this piezo 1 and TOR interaction.
No, actually, it's already internalized. You know,
I see.
But what we show actually it's we check this toll-like receptor is not on surface. Actually, it's already on the. Oh, I forgot that to answer before the Sarah's question. So actually, what we check is already inside the cells. So you can see we use the LPS coded beads. So it's engrafted, and then this like the membrane wrapped on these beads, and then toll-like receptor is located on those engrafted beads, and then we can also detect the piezo one with them. And for the E. coli also, like once it's engrafted, and then we can check the toll-like receptor and piezo one, both localized on this engrafted bacteria. So before I forgot to say that. Yeah, it's not really on the cell surface. Yeah.
But if it's not, then my next question is, the toll-like receptor will interact with the piezo on the surface, so it will induce calcium influx, right?
Right. So
this interaction only will happen when the toll-like receptor get activated.
Yeah, I think that's like you guys' question really making me thinking clearly. So I think that it's probably because for calcium, even for the opening of the piezo 1 receptor, it's transient. So I feel like once the LPS, it's because we just try to show that toll-like receptor and piezo 1 is colocalized, we have to do that. But actually, we some figures we didn't really put here. Actually, we can also see like on the surface for some cells, because the reviewer really asked us to do a lot of different staining. So even with the membrane, other proteins. So we did see like on the surface also can colocalize, but probably it's transient and then it's internalized soon. So we really didn't detect that. But for the calcium influx, it means that once your LPS is treated and then calcium influx, I think this influx it's enough to trigger the downstream signaling, because later our calcium influx assay showing the LPS treated and then rapidly like the calcium influx that wave, but it's diminished in the piezo 1 knockout cells. So I think it has function once the toll-like signaling triggered. But here we just want to show like it try to clearly show they are associated together. Yeah.
This is not my question. My question is, do you think the piezo and then toll-like receptor interaction only happen when the toll-like receptor get activated?
Oh, this is a very good question, because I think it will enhance the association, but we did see some weak colocalization in the control cells. So we also quite puzzled, but we were thinking maybe sometime once you culture the cells, somehow it's activated, or it just real like that. Yeah, we did see some weak colocalization when it's not stimulated. But after stimulation, the association or it's more condensed. Yeah.
So if that's the case, I guess my next question is, because I saw in your in vitro data, when you treat the cells with LPS, you see a phosphorylation of MOB1, which is activation of MST1 and 2.
Yeah.
And if you have piezo-1 knockout, then this phosphorylation looks like complete gone.
Right. Yeah, I also saw that data. So I was
This is actually quite this is really surprising, because I know TOL4 receptor can activate a lot of iron channel.
Yeah. Yeah.
The dominant calcium influx, at least I know the TRP channel can really induce a lot of calcium influx. Now, if you say the piezo knockout can completely block the calcium influx, that's actually quite surprising. Where the other contribution of the other?
No. We didn't say that it's completely blocked, because the Yoda one is piezo-1 specific agonist. So if piezo-1 knockout, we completely block the calcium influx. But for LPS, no. We still can see halfway of calcium influx. You can see these figures. Yeah. Oh, sorry. So here. But I did yeah.
No,
yes.
calcium influx, I know it's not complete block. I'm asking for the MOB1 phosphorylation.
MOB1 phosphorylation.
If you knock out piezo, the MOB1 phosphorylation stimulated by LPS is completely gone or just a half gone? I forgot.
From our data, I think it's really decreased a lot. But I don't know whether it's a specific piezo MST1 kinase. It depends on the piezo-1 mediated calcium influx. I don't know whether actually I'm not very sure how to answer your question. But I will say that actually okay, hold on. Okay, here. So that's your question, right? So actually, you can see here for the phosphor P38 and phosphor junk actually completely not affected. So actually, we were thinking about that. It probably some very special activation mechanism of MST1 kinase, because we did see like you know knocking off out of piezo-1 totally could not affect other like a MAP kinase activity. So I know what you mean, but it could be like some special piezo-1 not okay. Not in another way, I would say that piezo-1 receptor through the calcium influx to activate the downstream chemogulin kinase 2 and then MST1 kinase. But it could not include whether piezo-1 itself can recruit other core molecules, some other molecules facilitate to activate MST1 kinase, not just dependent on the calcium. It could be calcium is the major effector. That's what my answer, because it seems like it totally cannot affect on the MAP kinase, but it did completely affect the MST1 kinase activity. So, but we try to identify some like other partners might depends on piezo-1 to activate. I think there's something, there's some molecules involved in that, not just depend on calcium, but calcium play a major role. That's my thinking, because I agree with your questions. We did thinking about this phenomenon. Yeah, LPS definitely not just induce the calcium for downstream signaling, not just depends on piezo-1.
Okay, thank you.
Yeah, thank you.
Okay, Lanfen, I got a few questions since there's no people on the list. Okay, so first of all, according to your data, it more looks like that the infection or the LPS incubation will bring the toll-like receptor 4 and the piezo-1 together, right? So I'm wondering whether there could be expression regulation for piezo-1, or it's only a change of the colocalization.
Oh, yeah, it's a very good question. Actually, we have that data upon the LPS or the bacterial infection, the piezo-1 expression level increased. increased in like it's really the protein level increased. We checked the mRNA level, yeah, increased.
So it depends on toll-like receptor for signaling, or it's not?
I don't know, but we just check because the basal level, the PSO1, but not PSO2 expressed on the macrophage. And we also check the mRNA level of PSO1 after the infection or after the LPS treatment. We did see the expression level of the PSO1 increased. So I'm thinking about, because like we just say that if you treated the macrophage, it really like promote the F-actin polymerization and cytoskeleton reorganization. And then I feel like once the cells, the stiffness or the physical situation changed, it probably it's a feedback or something triggered that those mechanical force receptor to express. That's what I'm thinking.
Okay.
But we did see the phenomenon. I don't know how actually the mechanism for the for increasing the expression. Yeah.
Okay, actually, I'm also very curious about how about these pathways like PSO1, MST1, and REC1 and actin polarization. Could they also regulate the FC receptor signaling, especially on the antibody-dependent cellular phagocytosis?
I don't know, but when we saw this phenomenon, we were thinking about, because PSO1 it's really very general receptors. It's not very specific. I mean, the general, I mean, it expressed everywhere, and then it has very general function, like sensing the environment. Like all the cells need this function, right? Need this function, sensing the environments, the pressure and also the shear power. It's required for all cells. So when we saw this the toll-like receptor and could associate with the PSO1, so actually what we're thinking about that PSO1 might be a very important co-receptor for many receptors, such as cytokine receptors, or maybe for you mentioned the FC receptor. I feel like it maybe it's an important like core molecules for those receptor. When the cell in the different environments, it will facilitate the cells the specific function. That's what we're thinking, but we really didn't check for that. But
It could be very interesting to explore the affecting. Yeah,
I feel like many receptor could be recruiting the PSO1 and to facilitate its function in response to the environmental stress.
Okay, thanks. So is there any other questions? Okay, if it's not, okay. So here it is. Thank you so much for Lanfen's great talk, and also especially it's amazing discussion after we almost spent like 40 minutes for the discussion. It's amazing. Okay, thanks Lanfen, and thanks for the organizers.
Okay,
thank you everyone.
Okay, bye-bye. Thank you.